structural computational biologist. She's focused on understanding the structure, function, relationship of macromolecular complexes. She has strong expertise in molecular dynamics and also in integrative modeling, and it's been serving as assessor in several rounds of the CASP challenge. And I leave her the stage.
Thank you very much, Anna. Also, thanks for the invitation. It's my first time here, and it's been, I really enjoyed it. So it's been absolute fun to be here. And thanks for the lovely introduction. So today, I'll try to talk about what has been happening in our field. So I'm a theoretician, like 100%. I have seen quite many experimental data presented here. Maybe it's going to be the first presentation without an experimental data, so we'll see how it goes. So in our field, we reached at a stage where our field became big data. So none of us could predict this to be happening, because we were always the low throughput science in the computational sciences, in the computational biology sciences. But now we have attended so many models, so many predicted structures, and now even extended to complexes deposited in the AlphaFold database, which are awaiting us to be analyzed. And there are really nice papers on it already. For example, there's this TED paper which has analyzed all the monomers, AlphaFold monomers available in the AlphaFold database, and is actually presenting which type of topological landscape we are dealing with in the UniProt sequences. So as I said, we could never imagine we would be reaching this point. So let me start with the background. So how did all start? So at the start of this, like sort of the origin of all these developments, you have the CASP initiation. So we have heard a couple of times during this conference, CASP. For the people who are not aware of what it is, it's a blind competition that has been taken has been happening since the last 30 years. It's a biennial competition. What is happening is that the experimentalists, namely most of you, you give your structures to the CASP committee, and then the predictors all around the world, they try to predict the structures. And for many years, this was only monomers. And then a handful of people from the community, they assess the state of the predictors. And every 2 years, we have papers on discussing the state of the field, etc. So CASP's first and main aim was to predict the 3D structures of the proteins. So they, at first, of course, posed it in a way that they want to predict the folding of the proteins. But now, after the release of AlphaFold, this has become something else. So here you see the plot that was released as a result of this AlphaFold revolution that happened in the 14th round of CASP in 2020. So here you see that in this. So here in this bit, you see that for the difficult targets for which there is no homology available in the literature, after the introduction of AlphaFold 2 in the competing teams, we see an accuracy that is within experimental error range. So it's the highest accuracy that has been ever reached up until this point. But when we investigate this plot, we note this extreme big success, but we also note something else. So we note that something happened over here around 2016, which actually made the field jump from one place to the other in the difficult cases category. And this concept was the use of coevolution. For many years, we used the consort columns in our function prediction. But then for a couple of decades, people have understood that actually if you have correlated evolution, this will indicate proximity in the structure. But it took many years until people could understand how to translate this into a mathematical expression or how to translate this into an AI model. And that was the time that actually needed for DeepMind to make the field jump from here to there. So in principle, what AlphaFold relies on is the input, mainly it's the input multiple sequence alignment. And its revolution is that it can link the multiple sequence alignment, templates as well if it's available, in one go to the structure. So that was the first layer of revolution. And the second layer of revolution is that it actually tells us it actually informs us on the accuracy of the structure, on the accuracy of the prediction. This we never had before, and we never imagined to have before, because all of our previous modeling scenarios, like most of them, they were physics-based. And this type of prediction you can only have if you're having an AI model. So this is how a monomer structure prediction field kind of jumped from one place to the other. But what happened in the complex prediction category? So this is the category that we are most interested in, because I'm working in this field already for a very long time. And thanks to this, I had the opportunity to serve as an assessor in the assembly category in 2 rounds of CASP. So, and I was lucky because in the 14th round of CASP, AlphaFold was out, but it was out for monomers. And in the 15th round of CASP, DeepMind did not participate, but everybody was using AlphaFold multimer to predict the complexes. So in a way, we observed the transition from more classical methods to more AlphaFold to revolutionary or innovative methods. And we observed the transition, and we observed the change of the success rate in the history of prediction of protein complexes. And this we obviously see here. So what we should concentrate on is basically this red part. This red part is the targets. Usually they are 40, 50 for each round. These are the targets for which the community did not perform well. So they are basically the wrong predictions. So if you see with the inclusion of AlphaFold 2, the unsuccess rate, or let's say, reduced from 60-something% to 10%. It was so amazing that when we were assessing the models, we of course don't know who is submitting the models. We thought that a new method emerged. We didn't think that it was simple AlphaFold 2. But then when the results are announced, and then when we know about the groups, we know about the methodologies, what we understood back then was, if you use AlphaFold at the time 2.2, so this was the latest version, without doing anything, you just submit your sequences into AlphaFold 2.2, and then you expect to get a response, then your success rate becomes 30% on the same amount of targets. But if you do some other things, which I'm going to talk about in the next slide, you can increase your success rate up to 90%. So this was very surprising for us. And that's why I always try to present this slide, because I think it might be also surprising for some other people as well, because we realized that these people, they increase the success rate in 2 ways. The first way is, so this is the top ranking group. The first way is that this group, they did not touch the sampling protocol of AlphaFold 2, but instead they curated their own multiple sequence alignment. So this is a sign that actually, if you have access to larger sequence databases than UniProt, your own metagenomics databases or whatsoever, you have a chance of getting a higher success in prediction. And then the second option is, you don't touch the MSA, you don't play with the MSA of AlphaFold. It's just standard MSA, but in the end, you start playing with the AlphaFold sampling parameters. What are these? You increase the number of seeds, but increase them substantially. Or you use different versions of AlphaFold. So interestingly, one would think like the latest version of AlphaFold should be the one that you should be using, but these are AI methods. They all behave differently. So in principle, using all AlphaFold versions available turned out to be the best strategy in predicting better structures. Also, there is this other extra option, which is activating dropout layers. And activating dropout layers means like basically incorporating some level of stochasticity into your sampling. And these are all very easy parameters to change. You even don't need to play with the code. But okay, to be able to do this type of exhaustive sampling, you need to run your sampling on a cluster, because, for example, Wollner, who tried this approach, they generated 6,000 models per complex. And somehow AlphaFold scoring worked, and they became successful. So when we investigated this 10% of the cases where we observed failure, we realized that these are all multiple sequence alignment related. So I think this is a very important point to take. If you are not very much experienced with the AlphaFold and related techniques, this is one thing to consider. If you don't have a good MSA, you're not going to get a good outcome. So in which type of cases we don't have a good MSA or a standard MSA? So in the case of antibody antigens, there is the type of evolution is just different than the other regular type of complexes that AlphaFold knows. And because of this, even if you have dense MSA for your target, it's not certain that you're going to get a good solution in the end. Or alternatively, design complexes, okay, they don't have an MSA basically because they're artificially designed, or the complexes that are having a higher evolution rate than the regular cases, like viral complexes, bacterial complexes. These ones are also not easy to predict. And another example is obviously is predicting multiple conformations from the same sequence. This is not easy as well. It's a different type of problem. Basically, you have one sequence, but multiple conformations satisfying your evolutionary restraints, let's say. And the other thing is that, of course, anything that is out of proteins coevolutionary standards, coevolutionary landscape is difficult to predict, like protein nucleic acid complexes, protein ligand complexes, post-translational modifications, everything. Because AlphaFold really learned how to translate this coevolution information that I mentioned into protein structures. So after these observations, and I'm sure that not only because of this, DeepMind was on to releasing the next version, but together with these observations as well, they released a version that tries to address all these problems that we observed in the 15th round of CASP. So for this, they completely updated the architecture of AlphaFold. It's a totally different model, totally different architecture. You can use it through a server, but you can also install it and run it locally as well. And by updating the architecture, now they are much faster, and they claim that some of the challenges are better addressed. So it's an amazing improvement, but even though I wasn't involved in CASP 16, I was, of course, having a close eye on it. What we observed was after the release of AlphaFold 3, the challenges stayed the same. They did not change. So we seem to have this little percentage which is challenging, but as you have seen, that little percentage is most of the biology. So, and that's the part that most of the computational people are slowly changing to work on. And during the remaining part of my talk, I'll present 2 projects that we are doing along this direction. So I'll present at first what we could do to model shallow MSA complexes, and in this instance, host parasite, human parasite interactions. And then I'll provide some clues on how we can use AlphaFold to predict, to get some insights or to predict flexibility signals. So we had an ongoing project already with a Colombian colleague, and they have bioinformatics predictions on several human-parasite interactions. So then I thought, okay, using AlphaFold would be a great opportunity to assign a confidence score to these predictions, because what else could we do? I mean, these are like orthology-based predictions, and they should have a lot of noise in them. And for this, learning from CASP, what we did was we used all the AlphaFold versions available to model these interactions. And then we only concentrated on the very confident scores. So in complexes, the confidence score of AlphaFold is made of PTM and IPTM. And it combines, if they're combined, given a value over 0.8, then we are on the good side. So that was our strategy. So of course, at first, we wanted to see which versions are producing which type of models, which type of confidences. And surprisingly, we observed that, okay, version 2, the version 2 class of AlphaFolds, they behave similarly, confidence scores wise. But strangely, AlphaFold 3 was ranking, like the confidence scores are more concentrated over lower confidences than higher ones. So this was very surprising for us, because 2.3 and 3, they have the same training regime, they saw the same structure. So we were curious to understand what's happening. Then what we did was we actually compared all the predictions we have one to the other. So we made a pairwise comparisons. For example, in this instance, we compared the same structure, same complex that is generated by 2.3 and 3. And what we strangely observed was the model was exactly the same, but the confidence was totally different. So this was so puzzling for us. We couldn't know what to do. And when we obtained this result, it was 224. It was in the year of 24. Sorry. So then I thought, okay, let's just write this up as a small report and put it out. Put it on bioRxiv. Maybe we'll get a feedback from the community. I mean, who knows? And then 2 days later, I received an email from the AlphaFold team asking for our data to reproduce what we had. And in the end, we have been exchanging a couple of emails. And then thanks to this coincidental result, we understand that AlphaFold 3 is a general model. And because it's a general model, whenever it thinks that something is missing in the system, it downscores it. So in this instance, for example, if you add different types of ions, this is a membrane protein, by the way, but it didn't work well with membranes. So if you add different types of ions, this is produced by the AlphaFold team, then you basically reproduce the confidence scores that are generated by AlphaFold 2.3. So this was very concerning for me. So to understand whether this is a general problem or this is only specific to our data set, what we did was we took CASP16 targets and we actually investigated their scores. And then in the end, there are 1 or 2 papers in the literature from Badra group that are also pointing towards similar direction. And in the end, I concluded that, okay, when you know that the interaction is happening, so it's a true interaction, AlphaFold 3 works, so you don't have this scoring problem. I mean, we would have realized it already if this would have been the case. But if you do want to have a profiling, if you do want to have a scanning within a pool of sequences that you have, then probably the system or the AI model that you want to choose would be one of the AlphaFold 2 models. And if you have the resources, all AlphaFold 2 models. So this would be the best way to choose models for experimental techniques, experimental testing. So in the end, scoring is still a challenge for the community. Us and others are working on it. But this is, I think, an important example that people should be aware of. That's why instead of presenting what we have been doing with our methods, I wanted to present this example so that you can be aware of how to use which methods under which circumstance. So in my second example, I'll try to demonstrate a prediction scenario where we hopefully have at least a dream or opportunity or a hint of linking flexibility with the sequence or sequence with the flexibility. So I already, during my PhD, I already worked with complexes that are experiencing hinge motion upon binding. So one of the chapters of my PhD was about dealing with these types of systems, predicting how to dock them. So always having this in the back of my mind, after having AlphaFold available, I started to think like, okay, if AlphaFold can fish out, it was not only me, there are other people in the community as well who are doing similar work. But if AlphaFold kind of found a way to link the MSA with the output, so if the proximity is there in the signal, so there might be also something in the ambiguity of the signal, if there's any ambiguity, which we could link with the flexibility. And this is already proposed in 2022 by another group. So the first version of AlphaFold, AlphaFold 1, which participated in CASP in the 13th round, which was scoring top, but it was not revolutionary. What they were doing is that instead of providing the final structure, they were providing distance probability distributions. So the first thing they thought was basically converting the multiple sequence alignment files into distance probability distributions for all the pairs, for all the C-beta pairs in your protein system. And then this is called histograms. And then in 2022, after the release of AlphaFold 1, a group of scientists, they reported that, okay, if you, for example, in your complex or in your monomer, if you have an open and closed state, you actually have both signals in your distance distribution. So in your histograms, you see two peaks, for example. Or if you have one single distance, it's a rigid monomer or it's a rigid complex, then you have a sharp single peak, which is really certain and guiding the sampling towards the sharp distance that is being suggested by the program. So after seeing this literature, I thought, okay, this is a perfect example to go back to what I did during my PhD. And this I pasted from our 2011 paper, which is still an ongoing question, obviously. So how can we predict which type of conformational change should be expected upon binding, so that we can be prepared for it? I mean, not only for computational people, but for also experimental people, it's a challenge to know whether you're going to experience a large conformational change upon binding or not. So it could have been great, of course, if there would have been a tool which would tell us, okay, you're going to have a hinge-based motion, or you're going to have a loop flexibility. So this type of information. And then I thought, okay, why not using histograms to try to predict this type of changes, because the uncertainty should be in the sequence, right? And for this, we started with a perfect test case, for which we already had the data, because it's a collaboration that we were involved in. So Rapp-Silberg group, they solved the structure of a mitochondrial complex between adenylate kinase 2 and AFM1. And what they had was they had this EM map, in which adenylate kinase 2 C-terminal tail is stacking into AFM1. So this was a never observed type of interaction before. It was novel. So even though they had the map and AlphaFold's prediction was fitting in the map, they were still like, okay, we are sure, but not so sure. Could you maybe do molecular dynamics on these systems? So then we did long-scale molecular dynamic simulations on the apo-adenylate kinase, on the complex adenylate kinase. And when we had this histogram idea, we said, okay, so there is this obvious change in the distance. So this is only 3 residue spanning range, which is changing totally such a short segment, but it's changing totally its distance upon complexation. So we thought, okay, can we see this in the histograms? But before going into this, we thought, what is the distance probability distribution that we can obtain from molecular dynamics simulations? So we already had the simulations, we drew the probability distributions, and then we realized that in the apo form, actually there is a slight, like a small bump, which is reflecting the closed distance. Okay, so then after having this is our baseline. So we wanted to compare this with the AlphaFold, with the histograms, distance probability distributions that we will deduce from AlphaFold. And we realized, I mean, I still cannot believe how this is happening, but we realized something amazing. So even the intensity of the probabilities were matching with the MD observed distributions. So this is, we have some other systems. This is a recently published paper. We have some other systems also in our paper, which shows that actually histograms has the potential to inform us on the type of the conformational change that we are going to observe. But even so, this is very preliminary. So we had the idea. We could demonstrate it very fast in a couple of cases, and then we wanted to have it out soon, quick. But in the end, so the amazing thing is that everybody was thinking about it. We have been thinking about it as well. Nobody has found the best way to deduce this. But obviously, even in the complex signals, multiple sequence alignment signals, there is a signal that from which we can predict certain types of dynamics. But then the second point is that obviously this is a very pinpointed example, and the other examples are also pinpointed. I knew that these are happening, these are going to be experiencing hinge-based motions, even though none of them are, except this one, in the training set of AlphaFold. But this also tells us that there is something, but we should find a way to extract this, because we cannot consistently get this signal properly. And for this, we need to have MD benchmarks. And this is also happening. So it's coming in the community as well. Maybe in a couple of years from now, we'll be presenting something more solid, building on the insight that I just gave you. So in the end, to finish, AlphaFold is the best tool that has ever happened to complex modeling field. I mean, it's really the case. But you have to know when to use it and when not to use it. That's very important. Also, you have to understand how its scoring function works. There are not so many papers about this, but hopefully there will be more in the upcoming future. But more and more people are noticing the behavior of these AI models. And for this, benchmarking is essential. So it's not maybe fun, but it is something that we have to do. And quite a number of people are working on it. And finally, hopefully soon, we'll have a better understanding of how we can link evolution to dynamics, basically get different signals from the same, different conformational signals from the same MSA. And with this, I would like to finish. We do a lot of different work, but I'm not gonna talk about them today. This is my team, and this is where we live. So right across Athens. If you happen to be around the neighborhood, just pass by. We are the only structural group there. So thank you.